Hello all, hope you're doing well today. Today we are talking about bioenergetics. This is a big chapter with a lot of information, so I've broken it down into a few different lectures to make it a little more palatable, and so here we go. As far as objectives, I expect for you to be able to understand the basic terminology of bioenergetics, what it is, and metabolism that's related to exercise and training. Be able to discuss the role of ATP and in muscular activity. Be able to explain the basic energy systems that are present in the human body and the ability of each to supply energy for various activities. Also be able to discuss the effects of training on bioenergetics of skeletal muscle and look at the substrates used by each energy system and discuss the patterns of substrate use with various types of activities. Be able to develop training programs that demonstrate an understanding of human bioenergetics and metabolism, and especially the metabolic specificity of training. Bioenergetics is the branch of biochemistry that focuses on how cells transform energy, often by producing, storing, or consuming adenosine triphosphate, or ATP. Bioenergetic processes such as cellular respiration are essential to most aspects of cellular metabolism, therefore to life itself. Metabolism is the process by which the body changes food and drink into energy. During this process, calories in food and drinks mix with oxygen to make the energy the body needs. Catabolism is what happens when you digest food and the molecules break down in the body for use as an energy. Large, complex molecules in the body are broken down into smaller, simpler ones. An example of catabolism is glycolysis. This process is almost the reverse of gluconeogenesis. Anabolism is the opposite of catabolism. It's the mechanism that takes smaller units like nutrients, cells, or amino acids and bonds them together to create bigger structures. One example of anabolism in action is when your body is trying to heal a cut. It adds tissue and structures around that wound. It also is the process involved in a child's growth and in building stronger muscles for bodybuilders. There are hormones associated with both processes, but that's a little bit beyond this conversation. Adenosine triphosphate is an organic compound that provides energy to provide many processes in living cells such as muscular contraction, muscle growth, and nerve impulse propagation. ATP is required for the activity of key enzymes involved in membrane excitability. You also have the sarcoplasmic reticulum calcium handling and myofilament cross-bridge cycling that's associated with it. The various metabolic pathways are regulated by a range of intramuscular and hormonal signals that influence enzyme activation and substrate availability, thus ensuring that the rate of ATP resynthesis is closely matched to the ATP demands of exercise. Regular training and various nutritional interventions have been used to enhance fatigue resistance via modulation of substrate availability and the effects of metabolic end products. Because the intramuscular stores of ATP are relatively small, they are unable to sustain contractile activity for extended periods of time. For example, during an all-out maximal exercise such as sprinting at a power output of 900 watts, which is roughly 300% your maximal oxygen uptake, or VO2 max, the estimated rate of ATP utilization could last less than 2 seconds if stored ATP were the sole energy source. The human body uses 3 types of molecules to yield the necessary energy to drive ATP synthesis: fat, proteins, and carbohydrates. Mitochondria are the main site for ATP synthesis in mammals, although some ATP is also synthesized in the cytoplasm. Carbohydrates are the most readily metabolized source of energy for the human body, ensuring our engines keep running. Containing around 4 calories per gram, a calorie is a unit of energy used in nutrition. Carbohydrates are a source of energy that is quickly metabolized into glucose. or aka blood sugar, which the body then burns as fuel. How quickly these carbs convert into glucose determines on how fast glucose levels rise in the blood. Depending on the kind and quantity of carbohydrate consumed, this rise in glucose can either be rapid or gradual, large or small. Fats are important for many essential functions in the body, aiding in hormonal production, nutrient transport, and inflammation reduction. Fats assist the body in functioning smoothly. 1 gram of fat contains roughly 9 calories and is the most calorie-dense member of the macronutrient family. Like carbohydrates, fats also provide fuel for the body and at rest are the principal source of energy. However, fats are not typically utilized until muscle, blood glucose, or glycogen stores are depleted, as is the case in fasted states or with exercise that exceeds an hour. While protein can be used for energy production in a process called gluconeogenesis, its primary functions are in cellular repair, hormone production, and pH balance, among others. However, this being said, eating more protein does not necessarily equate to gaining more muscle mass. In fact, your body can typically only process around 20 grams of protein in one sitting. Like carbohydrates, protein also takes in around 4 calories per gram. Bioenergetics is the flow of energy, a biological system, and describes the conversion of macronutrients, carbs, protein, fat, which again all contain chemical energy, into micronutrients of glucose or ATP, which can be used for energy. The energy is harvested from the breakdowns of chemical bonds within the macronutrients. Energy stored in the chemical bonds of ATP is used to power muscular activity. The ATP stored in skeletal muscle is replenished by 3 basic systems: the phosphagen, glycolytic, and oxidative, which we'll talk about in a bit. ATP is required for all muscular activity and growth. Therefore, it's important to understand how exercise affects ATP hydrolysis and resynthesis. The breakdown of one ATP molecule to yield energy is called hydrolysis because it requires 1 molecule of water. The catabolism of ATP is facilitated by the enzyme ATPase. Notice how the chemical reaction has arrows going in both directions, which means this is the reaction for both hydrolysis as well as resynthesis of ATP. So you have ATP and a water molecule, and with the help of ATP will become adenosine diphosphate, an inorganic phosphate, hydrogen, and energy. ATP is replenished in the muscle cells by 3 different processes. As we mentioned, the phosphagen system, glycolysis, and the oxidative system. The phosphagen system and glycolysis do not require oxygen and are therefore considered anaerobic and occur in the sarcoplasm of the cell, while the oxidative system is aerobic and requires oxygen and occurs in the mitochondria. Out of the 3 macronutrients, only carbohydrates can be metabolized for energy without oxygen, therefore making it essential for anaerobic metabolism. The way this section is taught is totally highlight the fact that all 3 energy systems are active at any given time. The magnitude of each system's contribution to overall work performance depends mostly on the intensity of the activity and also on the duration of the activity. Those are our key points that will come up repeatedly throughout this chapter. So really important that you get that down. Therefore, other metabolic pathways must be activated, including substrate level phosphorylation or anaerobic and oxidative phosphorylation or aerobic. This is a great chart. I recommend that you take a picture of this, whatever you have to do. It's got great information, it is really symptomatic, and I think it's something that you're going to need for later on in your exercise physiology class and other materials. During exercise, the body relies on different energy systems to provide the necessary energy for muscular contraction. The three main energy systems involved in exercise are the immediate phosphagen system, the anaerobic glycolytic system, and the aerobic system. Let's take a closer look at each of these energy systems. So the anaerobic or glycolytic system is anaerobic by nature, again, meaning it does not require oxygen, and it provides energy for activities lasting from a few seconds to a few minutes. It primarily relies on the breakdown of glucose stored in the muscles and glycogen or in glucose circulating in the bloodstream. The process known as glycolysis involves the breakdown of glucose to produce ATP. However, glycolysis does not require oxygen and is considered anaerobic. The system can provide energy at a faster rate than the aerobic system, but generates lactic acid as a byproduct, leading to muscle fatigue. Activities such as sprinting, intense weightlifting, and high-intensity interval training primarily rely on the anaerobic system. The aerobic system, on the other hand, is a primary system for long-duration, lower-intensity activities. It relies on oxygen to produce ATP through the breakdown of carbohydrates, fats, and, to a lesser extent, proteins. In the presence of oxygen, glucose can be fully oxidated through a process called aerobic glycolysis, producing a larger amount of ATP compared to anaerobic glycolysis. Additionally, fat stored in adipose tissue and muscle can be oxidized to produce ATP through a process called beta oxidation. The aerobic system is efficient but slower in generating ATP compared to the immediate and anaerobic systems. Activities such as long-distance running, cycling, swimming, and endurance events primarily rely on the aerobic system. During exercise, the intensity and duration of the activity determine which energy systems are predominantly used. Short and high-intensity activities rely more on the immediate and anaerobic systems, while longer and lower intensity activities rely more on the aerobic system. However, it's important to note that all three energy systems contribute to energy production simultaneously, but their relative contributions vary based on the exercise intensity and duration. Training and conditioning can also affect the efficiency and capacity of these energy systems, improving overall exercise performance. The immediate phosphagen system provides energy for short duration, high intensity activities. It relies on stored ATP or adenosine triphosphate in the muscles, which can be rapidly broken down to release energy. However, the ATP stores in the muscles are limited and it can only sustain activity for a few seconds. To replace ATP stores, the phosphagen system also utilizes creatine phosphate, which is a high energy compound stored in the muscles. Creatine phosphate can quickly donate its phosphate group to ADP or adenosine diphosphate to regenerate ATP. This system is predominant during activities like short sprints, powerlifting, and high-intensity weightlifting that we talked about. And you can see it's a system that only lasts for about 6 to 10 seconds. The anaerobic energy pathways have a much higher power or rate of ATP production, but a smaller capacity, looking at total ATP produced, than the aerobic pathways. During very intense efforts lasting seconds, such as throws, jumps, or 100 to 400 meter sprints, or during intermittent game activities and field sports, most ATP is derived from the breakdown of phosphocreatine or PCR and glycogen to lactate. Direct measurements of muscle PCR and glycogen before and during after such exercise bouts show substantial decreases in the level of these substrates. The decrease is generally greater in type 2 than type 1 muscle fibers. The muscle ATP concentration is reasonably well maintained, although it may decrease around 20% during very intense exercise. The large increases in ATP utilization and glycolysis, as well as the strong ion fluxes during such exercise, result in metabolic acidosis. After the exercise duration extends beyond approximately 1 minute, for example, an 800-meter track event, oxidative phosphorylation is the major ATP generating pathway, and intramuscular glycogen is the dominant fuel source. Although it's relatively less studied, resistance exercise as seen during lifting events is also associated with substantial metabolic perturbations and contracting skeletal muscle. Now, creatine phosphate, or PCR, is an energy reserve for rapidly replenishing ATP and is stored in limited amounts in the muscle cells. Creatine kinase is the enzyme that catalyzes the reaction. This process is active at the start of all exercises during the first 0 to 6 seconds and is mostly used for short-term, high-intensity activities. Creatine kinase breaks down creatine phosphate, steals that inorganic phosphate, and produces energy. Then the energy then adds the phosphate to ADP to synthesize ATP. Anaerobic exercise is a type of exercise that breaks down glucose in the body without oxygen, meaning it's anaerobic. In practical terms, that means that anaerobic exercise is more intense but shorter in duration than aerobic exercise. Biochemistry of anaerobic exercise involves a process called glycolysis, in which glucose is converted to adenosine triphosphate, or ATP, which is the primary source of energy for cellular reactions. Lactic acid is produced at an increased rate during anaerobic exercise, causing it to build up quickly. That anaerobic exercise may be used to help build endurance, muscle strength, and power. So glycolysis is the breakdown of carbs to resynthesize ATP. There's two sources of carbs in the body used for glycolysis, and muscle glycogen stores and glucose from the blood. The glycolysis process is driven by many enzymatic reactions and therefore takes a longer time to resynthesize ATP than the phosphagen system, which only requires one enzyme. But glycolysis has a much higher capacity to produce ATP because of the abundance of glycogen and glucose. Both glycolysis and the phosphagen system occur in the sarcoplasm and are considered anaerobic. Primary source of ATP for high intensity activity up to 2 minutes. The resynthesis of ATP via breakdown of PCR buffers some of the hydrogen ions formed as a result of ATP hydrolysis. The PCR in muscle is immediately available at the onset of exercise and can be used to resynthesize ATP at a very high rate. The high rate of energy transfer corresponds to the ability to produce a high power output. The major advantage of this system is its limited capacity. The total amount of energy available is small, and if no other energy source is available to the muscle, fatigue will occur rapidly. An additional pathway to regenerate ATP when ATP and PCR stores are depleted is through a kinase reaction that utilizes two modes. Pyruvate is the end result of glycolysis and can be dealt with in two different ways, depending on the intensity of the exercise. Although the name suggests the two processes depend on oxygen availability, it's not necessarily true because glycolysis itself does not depend on it and is confusing and honest, rude that someone named it such. Anyway, glycolysis itself, is anaerobic. It's not depending on oxygen, but pyruvate is handled differently depending on the availability of oxygen and the energy demands of the exercise. So anaerobic glycolysis, or fast glycolysis, is used with high intensity, high energy demand, but it has insufficient oxygen. So pyruvate is converted into lactate. Anaerobic glycolysis assumes a major responsibility for the provision of energy and results in the production of a large amount of lactic acid and lowered pH in the sarcoplasm, which is believed to eventually hinder force and power development by the muscle fibers. Lactic acid values in the blood rise commensurate with muscle contractions. If demand is high and intensity is high, energy must be transferred more quickly The body will depend on anaerobic glycolysis and convert pyruvate into lactate. On the other hand, if energy demands and exercise intensity are low enough and enough oxygen is available, the body can shuttle pyruvate to the mitochondria to be oxidized. There are two main processes available for regeneration of nicotinamide adenine dinucleotide, or NAD+, in muscle. Reduction of pyruvate to lactate will achieve this, and this reaction has the advantage that it can proceed in the absence of oxygen. Lactate can accumulate within the muscle fibers, reaching much higher concentrations than those reached by anti-glycolytic intermediates. But when this happens, the associated hydroions cause intracellular pH to fall, and some lactate will diffuse into the cellular space and will eventually begin to accumulate in the blood. Aerobic glycolysis is slow glycolysis, and it really works with more of lower intensity exercise, a lower energy demand. It has sufficient oxygen, so pyruvate is shuttled into the mitochondria and converted to acetyl-CoA and enters the Krebs cycle, also known as the citric acid cycle, which we will talk about later. The byproduct of anaerobic glycolysis, which is lactate, has traditionally been thought to be detrimental to muscle function. However, it appears likely when lactate levels are very high. Elevated lactate levels are only one of many changes that occur within and around muscle cells during intense exercise that can lead to fatigue. Fatigue, that is muscle failure, is a complex subject that depends on more than just changes to lactate concentration. Energy availability, oxygen delivery, perception of pain, and other psychological factors all contribute to muscular fatigue. Elevated muscle and blood lactate concentrations are a natural consequence of any physical exertion. The effectiveness of anaerobic activity can be improved through training. Anaerobic exercise also increases an individual's basal metabolic rate, or BMR. Some examples of anaerobic exercises include sprints, high-intensity interval training, and strength training. Anaerobic metabolism uses glucose, as we've talked about, as its only source of fuel and produces pyruvate and lactate acid. Pyruvate can then be used as fuel for aerobic metabolism. Lactic acid can be converted back to pyruvate in well-oxygenated muscle cells. However, during exercise, the focus is on maintaining muscle activity. Lactic acid is transported to the liver, where it can be stored prior to conversion to glucose in the presence of oxygen via the Cori cycle. The amount of oxygen required to restore the lactic acid balance is often referred to as the oxygen debt. Despite activation of the oxidative pathways in skeletal muscle during exercise, accelerated rates of glycolysis result in the production of lactate, which accumulates in muscle and blood, particularly at high exercise intensities. Although lactate was considered simply a metabolic waste product for many years, it's now recognized as an important substrate for oxidative metabolism, gluconeogenesis, and muscle gluconeogenesis, and as a signaling molecule mediating exercise adaptations and interorgan communication. In red blood cells and other tissues that lack mitochondria and the exercising muscle tissue that favors anaerobic metabolism, glucose is converted to pyruvate and subsequently to lactate. Lactate is secreted into plasma and picked up by the liver for conversion into glucose via the Cori cycle and via a redox reaction catalyzed by lactate dehydrogenase. Now, gluconeogenesis is a metabolic pathway that results in the generation of glucose from certain non-carbohydrate carbon substrates. For gluconeogenesis to occur, the ADP and ATP ratio must be very low, since gluconeogenesis is an energy-demanding process, requiring high energy molecules to be spent in several steps. Now, lactate formation can occur during aerobic conditions as well, and lactate production is a result of glucose utilization by muscle cells under aerobic conditions. Lactate is not necessarily a waste product. In fact, it is the most important glucogenic precursor or new glucose generator in the body. About 30% of all glucose we use during exercise is derived from lactate recycling to glucose. Lactate is also a key regulator of intermediary metabolism, regulating substrate utilization. It decreases and inhibits the breakdown of fat for energy purposes, or lipolysis, as well as the rate of glucose utilization by cells, which is called glucolysis. Lactate is the byproduct of glucose utilized by the muscle cells, and so the higher glucose flux into the cell, the higher the lactate production, independently of oxygen availability. So during your high intensity exercise, your type 2 fast muscle fibers are fully recruited due to high contractile demands by skeletal muscle to produce energy, or ATP. Your type 2 muscle fibers are highly glycolytic. They use lots of glucose, which results in the production of high amounts of lactate. This production is a natural byproduct of glucose utilization by the skeletal muscle cells. So during intense exercise, lactate production is many times higher than that of resting levels. The release of hydrogen ions associated with lactate can cause an important reduction of contractile muscle pH, resulting in acidosis. This excessive accumulation of hydrogen, not only from lactate, but also from the ATP breakdown for muscle contraction, or ATP hydrolysis, may interfere with muscle contraction at different sites. For example, it may compete with calcium for troponin C binding site, a protein involved in muscle contraction regulation. Hydrogen may also inhibit calcium release and reuptake from sarcoplasmic reticulum. Both processes are involved in muscle contraction. All this can result in a decreased muscle contraction capacity, which can cause an important decrease in peak twitch force, a decrease in maximum muscle shortening velocity and performance. Lower blood lactate levels observed in top athletes is due to an enhanced lactate clearance capacity. Lactate can be exported to the blood for clearance and energy purposes in pretty much every organ in the body. However, this process takes time, minutes, while lactate is produced continuously during exercise. Well-trained athletes are very efficient and export less lactate to the blood as they clear it in higher amounts, right in the lactate-producing muscle, which takes seconds or milliseconds. It's very advantageous as it allows contractile muscles a faster hydrogen removal, as well as a faster lactate recycling for extra energy ATP. During exercise, lactate is mainly produced in fast-twitch muscle fibers, which use lots of glucose for energy, and is cleared mainly by slow-twitch muscle fibers. It's a complex process involving different lactate-specific transporters and enzymes. Fast-twitch fibers have a high content of one transporter called MCT4, or monocarboxylate 4, which transports lactate away from the fibers. Slow-twitch fibers possess a transporter called MCT1, which takes lactate inside the fibers. Lactate training, also known as lactate threshold training or anaerobic threshold training, is a training method used to improve an athlete's ability to tolerate and clear lactic acid from muscles. Lactic acid is a byproduct of anaerobic metabolism that accumulates in the muscles during intense exercise, and it's associated with muscle fatigue and limits performance. Lactate training aims to raise the lactate threshold, which is the point at which lactic acid begins to accumulate in the muscles faster than it can be cleared. By training at or near this threshold, athletes can improve their ability to sustain high-intensity efforts for longer periods of time. So lactate training involves exercising at a high intensity, typically around the lactate threshold, usually around 80 to 90% of an individual's maximum heart rate, or at a pace that feels somewhat hard or challenging. Interval training is commonly used in lactate training. It involves alternate periods of high intensity exercise near the lactate thresholds with periods of low intensity activity recovery. Sounds a little bit like HIIT, right? Like other training methods, lactate training involves gradually increasing the intensity and duration of workouts over time to provide a progressive overload stimulus to the body. This helps to improve the body's ability to clear lactate and sustain high-intensity efforts. Lactate training should be tailored to an individual's fitness level, goals, and sport-specific demands. Athletes may undergo lactate testing to determine their lactate threshold and establish appropriate training zones. Benefits include improved endurance, increased lactate threshold, enhanced anaerobic capacity, and reduced fatigue during high-intensity exercise. It is commonly utilized by endurance athletes such as runners, cyclers, and rowers, but can be beneficial for athletes in other sports that require bursts of high-intensity effort. And that is all that I have for you today. I look forward to talking with you again. Chapter 3 has a lot of information. So we will be continuing our conversation in bioenergetics. I look forward to talking with you again. Have a great day.